ideas, experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios.
Muy buenas tardes. Dos. Muy buenas tardes. Y para cerrar y concluir con los trabajos del Congreso Interpolitécnico de Investigación para alumnos de posgrado, damos paso a la última conferencia magistral denominada Regulación de anticuerpos en vacunas y cáncer por la doctora Isabela Pedrosa de Nuffield Department of Medicine de la Universidad de Oxford, de la cual me permitiré leer una breve semblanza. La doctora Pedrosa ha realizado importantes contribuciones en el campo del VIH y la vacunología, desde revelar los mecanismos celulares que regulan las respuestas de las células B y las células T, foliculares auxiliares, hasta descubrir las características celulares predictivas necesarias en las estrategias de vacunación para desarrollar anticuerpos neutralizantes amplios y potentes. Mediante la aplicación de nuevas tecnologías y el desarrollo de metodologías que evalúan exhaustivamente las variaciones genéticas y proteicas que dan forma a la función celular, el objetivo general del laboratorio de la doctora Pedrosa es mejorar nuestra comprensión de los mecanismos celulares y moleculares subyacentes a la regulación de las respuestas del centro germinal en la vacunación, la infección y el cáncer, para analizar objetivos mecanicistas que podrían usarse para mejorar las vacunas y las estrategias terapéuticas. Doctora Pedrosa, para los politécnicos es un gusto contar con su presencia y mucho nos agradará escuchar su interesante ponencia. Adelante, por favor, la recibimos con un fuerte aplauso.
Thank you for the kind introduction and especially for the opportunity to be here and to present our work in such a beautiful venue here at Politecnico. And as it was mentioned, my research is based on we're the Laboratory for Functional Immunology. We're very interested in the cellular and molecular mechanisms that drive and modulate antibody responses from the context of vaccination, infection, and cancer. And today, I hope that I can give you an overview of what are these mechanisms that are critical for the formation of antibodies that are specific to really tackle infection and also cancer. So the first part of my talk, I'm going to describe the role of germinal centers in secondary lymphoid organs. Then we will discuss the phenotypic and functional role of tertiary lymphoid structures, especially in cancer. These are structures that are being observed in very close proximity to tumors that seem to be associated with good prognosis. And that leads me to my third point, that is to talk about how we can determine their potential of these structures in terms of clinical relevance and to improve the current vaccines that we have, vaccines and also therapeutics. So just a little bit of an introduction to immunology. We all know that the immune system is a complex network of cells and molecules that defend the body against infection and various different infections that range from bacteria and viruses. So the immune system needs to be really broad to be able to tackle all these responses. And this is a coordinated effect by many cell subsets and really two different arms of the immunity. The first one is the innate immunity that it focuses on macrophages, neutrophils, NK cells, intrinsic immunity, and also complement. This is an arm of immunity that does not require picking up the pathogens. It is fast, it is strong, it just targets the response. And then after 7 days of the response, then we have, there's a cytokine storm and antigen presentation of these particular peptides that drive an adaptive response that is orchestrated by two main cell subsets, the T cells and the B cells. And the T cells can be subcharacterized into CD4 and CD8 T cells, and you probably are all familiar with the CD8 T cells that are very good at killing tumor cells and other components that are, and other cells that are infected as well. But also the B cells in the context of vaccination. And we know from SARS-CoV-2, B cells play a very important arm in mounting a specific antibody that can be just expanded and then tackle and lyse the disease or neutralize the disease in the context of infection. So the features of adaptive immunity is, well, we have 4 features. The first one is specificity. Our body has the ability to differentiate between different types of pathogens, whether it's a harmful or not. We call it self and non-self. It needs to be diverse enough to be able to detect the varieties of pathogens from bacteria, from protozoa and viruses. Also, as I mentioned, differentiate between what is self and what is non-self to avoid exacerbated responses and autoreactivity. And this is one of the problems when we have responses that are not checked by the cellular mechanisms. And finally, memory. Memory is an important mechanism of the especially the adaptive immune response to be protected against repeated exposures to the same antigens. And this is where sequential vaccinations play a very important role, for instance, in SARS-CoV-2 that we are all familiar in the last 4 years. So when we then further stratify an adaptive response, it really boils down into two main components. The primary, which is a relatively fast response that happens, as I mentioned, after 7 days. And in the context of antibodies, you start seeing a very specific class of antibodies, which are called IgM, and the very, very strong IgG antibodies that are highly functional. And these responses can last up to 35 days after the first exposure to the antigen. And then we have secondary immune responses, and this happens when there's a second exposure to the same antigen, when we get infected with SARS-CoV-2 again. We already have a mechanism or a memory response from the B cells that allows a very rapid increase of antibodies of IgG that are really functional, and that goes again as a wave of IgM. So as you can see, our immune system is really armed from every single element in terms of having a response that is fast and having a response that is sustained, that really is the core of immunity. So the key differences in antibody production, for instance, in the context of vaccination, antibody levels and especially the affinity of these antibodies to bind to the pathogens increase with repeated immunizations. And so here in vaccination, we're actually emulating a secondary response to really have a sustained response that lasts for a long time. And this increase in antibodies is mostly seen with IgG as well as IgA and IgE, and is secreted by the so-called switch memory B cells that are capable to undergo somatic hypermutation and affinity maturation. So what are these main 2 processes, and why are they so important? Why do we care so much when we actually develop vaccines or we go into the cancer vaccines? And I like to put this video from the University of Toronto, and I don't know if you can hear the video, but this is, can you put the video, the sound a little bit up? Okay, don't worry, I can explain that. So you pretty much see a B cell. This is a pathogen that is captured by one of the BCRs of the B cell. In red, you can see the T cells that are actually at the first instat in the secondary lymphoid organs. Normally, this is underneath our arm peaks. You can see the presentation of these pathogens. Then these B cells, it is not enough the affinity that they have, so they actually migrate into these kind of clusters or kind of, it's a racing of grapes, where they actually acquire point mutations in one of their BDJ regions that acquire this affinity maturation. And this will become more clear. Actually, here, this is one of the antibodies, and you can see these very light regions where point mutations are incorporated in a very massive rate. And this leads to the generation of a diverse pool of antibodies with varying affinities for this particular antigen. And then you can see here as this an actual structure of the antibody, where you can see these loops in red that are actually a change in terms of size over time that also define whether the antibody has a high affinity for a specific pathogen or not. Then we come, so after these point mutations as well, it's also very important that the affinity and the availability of the antigen that is presented in this particular secondary lymphoid organs actually drive the fate of a specific B cells. And we have 3 different scenarios in this case. We have a B cell that does not have an affinity for a BCR. In this particular context, it then goes to apoptosis and dies. If there's a decrease in affinity of BCR, it doesn't have the strong enough signal to actually be maintained, and also gets lysed and released then. But only the B cells that have an increased affinity of BCR actually survive, and those are the ones that are highly, they undergo into blast differentiation, and they secrete a lot of antibodies that are important for the therapeutic strategies. So you're probably familiar as well with all the antibody isotypes, and we have 5 different classes that are actually ranged across the distribution in the body. So mainly in our gut, for instance, we have predominant for IgA to actually react against commensal microbiome. And to have tolerance mechanisms in check. But also IgG is actually one of the isotypes that as vaccinologists we're most interested in because of the properties of highest opsonization and neutralization activities that I had already alluded in the previous slides. But we also have IgM with a different structure that's produced first upon antigen invasion and increases transiently, and as well, so IgD and IgE where we have that is involved in allergy. So I explained so far where are these antibodies, what type of mutations do they have, what type of affinity maturation, also they're highly dependent on the concentration of antigens. That is also very important from the context of vaccination, but where does this affinity maturation occur? And this actually happens in the lymphoid organs. So the lymphoid organs are a number of morphologically and functionally diverse organs and tissues that have different functions, and they are classified as primary and secondary. So now the thymus and the bone marrow are the primary or central lymphoid organs where actually maturation of mostly T-cell lymphocytes takes place. But we also have a set of secondary lymphoid organs that include the lymph nodes, the spleen, and various mucosal-associated lymphoid tissues, such as gut-associated tissue in our gut microbiome. In our gut, which functions to trap antigen and provide sites for maturation to interact with specific antigens that we need to mount a response against. Now, in these particular lymphoid organs, then we have what we call the lymph nodes. And the lymph nodes are a very small group of small bean-shaped organs that are normally around 2.6 centimeters in length, as you can see here in the histology section. And they're found mainly in the neck and trunk of the human body. And as you can see, for instance, when you get a vaccination, you get swollen lymph nodes. This is actually what's happening. You're mounting a response, your T cells and your B cells are interacting there with the specific vaccine or an immunogen, and then you mount an adaptive response that is important for antibody responses. And as they play actually a vital role in the filtration of antigens and debris from the lymph nodes and in the generation of immune responses to specific pathogens. And you can see here that as the lymph nodes really are hubs to mount humoral responses. You can see that and here in the secondary lymphoid organ, how the B cells and the T cells really go into this kind of particular pockets where they interact and they form and they come in via these high endothelial venules, which are really kind of like pipes or vessels that actually allowed migration of T cells and B cells. And then what you can see here is a germinal center reaction where actually T cells and B cells spatially locate strategically with each other to actually for T cells to help B cells undergo these somatic hypermutation and affinity maturations that I have described in the previous slide. So the germinal center reaction is actually not only a small pocket, but it's actually highly compartmentalized. And here in this graphic, it really depicts what happens with an activated B cell to give rise to these old centroblasts that have that start acquiring these point mutations. And then there's these different fates of the CDL of the B cell. So one where there's no information from the pathogen, so it goes into apoptosis. Other cells, they interact with another cell type, which is called follicular dendritic cells, which also capture antibodies I'm going to show in the next slides. And they go into memory B cells. They are not as high in affinity as opposed to when B cells actually interact with the specialized T cells and then undergo all these processes and turn into plasmablasts and plasma cells that secrete a lot of antibodies. So the follicular dendritic cells, these are actually, they're really important in the generation of these germinal center responses because they bind the immune complexes, as you can see here, in the tentacles, in a way, with native antigens. For instance, they bind intact proteins, and they can also express native antigens in a very large, considerable amount of times for many years, especially in humans. And they provide a very important source of antigens to the B cells. Another very important T cell subset that we have been studied through the years, both in mouse and in humans, as a collective scientific community, is actually a CD4 T follicular helper cell. So CD4 T cells come in various different flavors. They have different functions, as you can see here. But actually, these T follicular helper cells, what they do is that they provide cognate interactions to the B cells in these germinal centers in a very specialized area, which is called the light zone, and then allow these B cells to go around in circles and then start acquiring affinity maturation to a very specific pathogen that is actually presented to the CD4 T cell. So this again is another, we call it a check, kind of a QC to allow good affinity antibodies to actually develop and to survive. And so the dynamics of these germinal center responses, that was as what we have seen in murine models and also in humans, is that it takes around 7 days to actually form where B cells, the follicular B cells, they start going into these primary follicles. You start seeing the presentation of T cells and B cells and an organization of this expanding germinal center in a dark zone that then resides after 14 days and so on. So how is this important in the context of vaccination, for instance? And this is work that I have done in the last 10 years as part of an NIH consortium where we were focused on, our main goal was to develop HIV vaccines. And as you know, we have been trying for 30 years plus to develop a vaccine that is efficacious, and we have not yet been as successful as compared to other vaccines like what we had in SARS-CoV-2. And the reason being is because of first of all, the structural properties of this, of the HIV. HIV is very good at hiding the proteins, so it's very difficult to get a candidate or an immunogen to develop in a particular vaccine. But most importantly, because the proteins that the HIV presents, they're often restricted to tolerance mechanism. They share proteins with self, and so this is the way how they hide, and a response of a B cell that is a precursor to develop this particular antibody is often deleted by our tolerance mechanisms, and this is why we never get an HIV vaccine, an antibody that is highly neutralizer. And so we know all this context from the viral perspective, but from an immune landscaper, we knew that there might be something going on. And the reason being was because after 2 or 3 years of infection, some people living with HIV actually were capable to develop these broad and potent neutralizing antibodies. They could serve regions of this HIV, and then we thought, okay, there must be something in our immune system in terms of regulation that we can learn from to actually emulate and put back in our immunogen designs. And this is exactly what we did. So we actually were lucky to have a cohort of more than 240 people living with HIV from whom we have neutralization data available, as you can see here in the plot. So this is all the range of neutralization in the x-axis. Sorry, the patients in the x-axis and the neutralization breadth on the y-axis. And so we thought, okay, if we want to understand the immune landscape, we need to go to the extreme ends of the cohorts. And so we selected the top and bottom quartiles and ended up with 50 people in each end, and then matched with demographically matched seronegative controls. And in the last, this is work that we actually did in 10 years, so this is kind of in a nutshell. We were able to really understand the immune landscape that is characterizing these particular patients. And what we thought is what we actually hypothesized from the beginning is that actually these patients have an altered balance between the so-called CD4 T follicular helper cells. They have more activated CD4 TFH, and also lower mechanisms of regulation, so all these immune cells that have the function of controlling proliferation of the CD4 T cells. And this is how we actually started building up all this information. So when we looked as a consortium, we found, okay, you know, these B cells have alterations in the IgG repertoire. They are actually, there's a high predominance of autoantibodies, so we knew there was reduced tolerance mechanisms. It's something work that we published in 2016. Then we looked at as I said, the activation of CD4 T follicular helper cells. And then we started looking at all these regulatory cells. We started with the very classical CD4 Treg cells. We knew they were dysfunctional. And then also we looked at NK cells. So this is a cell subset that is part of our innate immune system. Also reduced. We knew that the NK cells can actually regulate all these processes. And finally, this is actually unpublished work where we shown that regulatory B cells are also dysfunctional. So all together, all these arms of tolerance mechanisms are lower. And this allows the CD4 T follicular helper cells to be more activated and to provide more help to the B cells to provide these very rare antibodies that are often deleted by tolerance mechanisms from the B cell perspective. And so we are actually very excited about this work because what we did is develop machine learning models because we had, with this information in a multi-omics approach, we ended up with more than 9,000 features from these particular people. And so we actually did the prediction models, and now we're happy to say that we have a signature that we're looking and we're quantifying in our preclinical and clinical studies and trying to design adjuvants to actually drive that path of neutralization to have a more efficacious HIV vaccine. So this is one example where understanding the biological and the cellular processes that regulate antibody responses in a very challenging scenario are helpful for vaccine strategies. Now, as a summary of this part, so I shown that what is a germinal center, the role of somatic hypermutation to have a better response, what affinity maturation means, the role of follicular dendritic cells and follicular helper T cells, as well as all the regulatory cell components. But then this is where we started being interested about cancer. So in the last 10 years, clinicians started observing that small lymphoid aggregates in the very close proximity to tumors. And every time that they were finding these structures, it seemed to be associated with good prognosis and actually better response. And so as a scientific community, we thought, okay, these look very similar to the germinal centers. They have B cells, they have T cells. There must be a local response that is happening that is actually worth looking at to do better, to do vaccines and to learn from that instead of looking at the whole tumor microenvironment. And so this is what we started to do. I mean, tertiary lymphostructures are actually not only present in cancer, they're all present in other autoimmune diseases, and this is because there's dysregulation of these responses in the local microenvironment, but as well in chronic inflammatory diseases and chronic kidney disease. And as you can see here, when we started looking more and more about these structures, we actually came to the realization that the formation, the shape, the composition of the structures were really different across patients. We were finding all these cell subsets, but we were finding immature TLS, what we call just the very small aggregates of T cells starting to interact with the B cells. But we started also seeing what we were thinking in terms of primary follicle like TLS or even secondary follicle TLS with also fibroblasts, so a more sophisticated and more well-structured mature TLS that was always associated with better response. And so this is where we started kind of systematically looking at these particular structures. And there's current techniques to characterize this TLS in its maturation. It's rather challenging actually to look at TLS because they're often rare. It really depends where the accessibility of your sections, where you cut as well. So one needs to be methodic and have a toolbox of methodologies that you can use to actually characterize this TLS. So the most common one actually comes from pathology. So this is an H&E where you can start seeing these dark purple blobs of what we think could be a potential tertiary lymphoid structures. And then we started, with the years, started being more really harnessing the technology in terms of going from immunohistochemistry to multiplex immunofluorescence, all the way to the latest spatial transcriptomics that allows us to characterize more than 5,000 genes, so 1/4 of our transcriptome, and in a single cell resolution. So we're very excited about starting to look at this because it will give us information about pathways and also trying to dissect the function of this particular TLS and the range in various different patients. All right, so far the community was, we have a TLS assessment from the H&E where you can quantify around 50 cells with a visible germinal center, as you can see here, which we call mature TLS. And then if there's, for instance, fewer cells but without a visible GC, we ask if they have the presence or the infiltration of follicular dendritic cells or different types of maturation of B cells as well, to call them mature TLS, immature TLS or no TLS at all. And then you can see all these cases where the inflammation is actually, there's no inflammation and there's no cell aggregates. And when we look at our multiplex immunofluorescence, you can actually start seeing work. I think this one is from breast cancer, where you really see this kind of very nice structure of a germinal center with CD8, CD4 T cells in the perivascular region of the tumor that is kind of on the side. And this is another example of, for instance, spatial transcriptomics, where this is actually 500 genes, where you can see this the whole H&E, and then you can zoom in really almost into 20 micrometers, and they're starting to characterize these particular structures. So, as I mentioned, these TLS are associated with better clinical prognosis in cancer, and it's really in most of the cancers. So we have seen these observations in oral cancer, breast cancer, lung, pancreatic, colorectal, melanoma, and ovarian cancer. In breast cancer, they're often very well defined. So you can see these germinal centers, specific T follicular helper cell, and also germinal center B cells in here. And in colorectal cancer, you see something a little bit more, in this particular case, more round, where you have CXCL13, which is a chemokine that sustains structures from what we know in the secondary lymphoid organs, but also have follicular dendritic cells and germinal centers. And when this particular lab actually quantified these particular cells, they actually saw that having at least one mature TLS, you have 70% less probability of recurrence after 3 years. So it's definitely looking at something that is meaningful from the immune perspective that is worth exploring. And this is not only in untreated patients. You can also see in checkpoint blockades, for instance, against CTLA4 and PD-1, where mature TLS are often associated with better response and favorable survival in pan cancer. So you can really see here that people who survive much longer, they have all this higher percentage of them have mature TLS, and this is significant up to 50 months after recruitment of these patients. And so what we think is happening, we know that they're there, but what do we think the function is? So we think from what we know in terms of B cells, it has to be an antibody-mediated effect, and there might be looking at an antibody that is looking at a specific tumor protein that is worth exploring. We know that the B cell, all the B cell elements and all the B cell subsets that are required for the formation of a germinal center in secondary lymphoid organs are definitely there in the tumor microenvironment, as you can see here on the table. So you can see naive B cells, activated B cells, memory, all the way to plasma cells, which we've shown in the slides. And there might be different functions of what's happening. So it can be, these antibodies can be binding to the cancer cells and triggering complement-dependent cytotoxicity by calling macrophages and NK cells from the innate and adaptive cell to actually lyse tumor cells. And then also there's direct antibody effects where there's neutralization of intracellular proteins by making the tumors render less fit. But also via transocytosis, which is latest data that has been reported, where IgA specifically is able to bind to this particular receptor. It goes into the cell, and then it fishes out proteins that are intracellular or even they show intracellular and then put them back so they're presented to the immune system so they are visible for responses. And this is really interesting from the perspective of therapeutic strategies in that sense. So the next question for us is, okay, is the response happening local or is it coming from secondary lymphoid organs? And I would say that the question is still out there. We're still trying to find technologies to actually address this. But so far, this is actually a new technology, which is called BCM-HD, which every dot represents, it's a 50 micrometer dot that represents between 1 or 5 cells, where you can actually sequence the BCRs and the TCRs. So it makes it very powerful in terms of quantifying clonal expansion of specific cells that are more likely giving you an anti-tumor response. And as you can see here, for instance, in what they shown in renal cancer, this mature TLS, it has high clonotypes. And when you look at the flavors of these clonotypes, there seems to be an expanding cloning here and expanded cloning here. So now the question, what we're trying to do is actually try to find out what these B cells and what these T cells are looking for. And when you look at the mutation count, so how many point mutations these B cells are acquiring, you can actually see that the patients that have the in the areas of the TLS is lower than in the tumor area. And this actually suggests that the germinal center B cells are actually interacting with what we think at TFH in this particular TLS, and they're migrating to these fibroblast tracts to actually reach tumor cells and then lyse cells. And there's evidence for that actually in this particular paper where they can measure their stain for IgG and also apoptosis. And they actually saw in close proximity apoptotic cells with these IgG plasma cells, so suggesting that there's functioning here. And this actually shows the same thing. So this is a section from a patient with TLS, and you can see that there's a lot of clipped caspase-3 indicating apoptosis as compared to the patient with the no TLS, and this is actually statistically significant when you quantify all these cells. And in here in the multiplex, you can really see the clipped caspase-3, the IgG present, and also the migration or the infiltration of macrophages based on the expression of C68. So they can trigger actually antibodies cytotoxicity, dependent cytotoxicity. Then when we look and then we quantify in terms of the TLS, so the percentage of IgG staining in these particular sections, and we stratify against high and low expressors, you can actually see that this is associated with good response. So definitely suggesting this provides evidence that there's an anti-tumor response that could be sustained in this particular TLS. And then most of the field, when we think about B cells, we actually think about antibodies that either neutralize pathogens or lyse or neutralize particular proteins, as I shown. But there's also this element of B cells presenting peptides to the T cells. And in the context of cancer immunology, especially in terms of therapeutic strategies, the CD8 T cells, the killer CD8 T cells, play a very important role. And so to this end, actually, a B cell that is there locally, ready to go with peptides that can be presented to both CD4s and CD8s can also be an arm of immunity that can be leveraged. And so within this context, you can see in here that they can present, and these particular candidates can be leveraged in terms of vaccines. But not only responses, B cells can mount responses that are specific, but also they can just act as a milieu to recruit cytokines, to have a permissive environment for CD8 T cells to be reinvigorated and to be able to kill much more efficiently than in a cytokine in a milieu that is desertic of an immune response. So as you can see here, that there's all these elements of how B cells can contribute to anti-tumor responses, and really we're starting to look at this in a more systematic way, and the question is, can we actually find candidates to be able to do vaccines? So in general, so we know that in terms of anti-tumor responses, there might be also a primary response that is relevant. So T cells and B cells that are prime in our lymph nodes, and then go on into our tertiary lymphoid structures, or TLS that actually start from the beginning there, ready to go, with the advantage that they can be much more readily active. And you can see here in the table the various differences on how well, the induction, for instance, secondary lymphoid organs are, they're developed during embryonic, whereas the TLS, they're there after birth. They're really, we still don't know what are the mechanisms that drive a TLS. So this is something that is very interesting from the scientific perspective, how we can actually do this or emulate the responses in patients. And the location, so the SLOs are in lymph nodes and spleen tones, Speier's patches in the gut, whereas the TLS are often in perivascular sites and also very close to the cancers. Development, they're pre-programmed, whereas the TLS are not. The capsule, you can see that in SLOs is really well defined within an environment, whereas the TLS might not be encapsulated and directly exposed to factors, which might be also beneficial from the anti-tumor response. The structure, they're highly compartmentalized T and B cell areas, whereas in the TLS, they're variable depending on the maturation stage. Maintenance, they're static versus transient. Self-tolerance, they're maintained, whereas in TLS, we think that there might be some TLS that are actually producing autoantibodies and they're disturbed. And in terms of function, they generate adaptive immune responses to the liver antigens, whereas in the TLS, they're responses to the locally presented antigens. So as a summary, in terms of the TLS, these are very active sites that are very attractive to understand from the therapeutic perspective in terms of generation of anti-tumor immunity, as they are locally, they're very close proximity to the tumor nets and neighboring tumor-associated antigens. And when we think this is all the whole picture of an environment that is sustained or that is contributed from the B cells, where you have an intratumoral mature TLS, your plasma cells actually migrate out through your fibroblasts, then you exert effector functions when you're calling other NK cells and macrophages to induce tumor cell death. Then you have also the arm of antigens and immune complexes that triggers that immune response. These antibodies migrate into the blood as well, go into and then reinvigorate as well the CD8 T cells. So this is why we think that actually after checkpoint blockade, when we trigger, we inject or when we infuse the checkpoint blockade to patients, there's an availability of neoantigens and apoptotic cells that is really great for the B cells to actually capture and present to the T cells. And this is why it's often correlated with checkpoint blockades. And in terms of the therapeutic approaches, well, understanding these anti-tumor responses can lead really to therapeutic strategies and not only therapeutic, but also cancer vaccines for prevention in people in high risk. So in summary, what we think is that if we can actually find candidates that are specific to tumor antigens or neoantigens that are not shared against healthy cells, then we can actually design vaccines using these tumor antigens that can be viral or messenger RNA or DNA or peptide-based, depending on any combination or whatever gives the best response. And then these APCs can take up the antigen, display these fragments on their surface. The B cells and T cells recognize these antigens and produce either antibodies and or killer T cells. And if tumor cells bearing the antigen develop, then these antibodies will be there and signal as a memory response to actually prevent lesions down the line. So that's our main overview in this context. So what are we doing in the lab? Specifically, we're very interested in colorectal cancer. Why colorectal? Because of the perspective of tolerance mechanisms in the gut. You can see we have various different tolerance in terms of regulatory T cells. We know that is important for regulation of humoral responses, so that's why we started looking there as well. And also because of the preliminary studies that suggest that the presence of mature TLS, it correlates with a reduced risk of recurrence after 3 years. And then we started asking. So this study is actually from a Caucasian, it's an Austrian cohort. And then we asked, can we actually see the same in the Mexican population? And this, the reason why we actually started looking at Mexico is because in terms of colon cancer cases, even though as compared to the UK, where we have almost 30,000 cases per year, whereas Mexico has 11,000, and the expectation of these cases will double by 2040, with 10% of the cases observed in young individuals. And we know that highest rates, that obesity plays a very important role in terms of colorectal cancer. And we know that Mexico, we do have a problem in terms of childhood obesity. So there is an important consideration from the public health perspective to actually better understand the anti-tumor responses, and especially TLS in the Mexican population as well, especially because we know in the gut microenvironment and what we eat heavily impacts on our immune responses. So I'm very delighted to say that we have started a collaboration with the Politécnico and also with the Hospital General, and that we have been building up in the last year, that really aims to understand these specific tertiary infrastructures in the Mexican population. And so we have a very talented people from Hospital General who are also gonna introduce the diet and nutrition component as well. We're gonna start collecting all this microbiome and fecal samples as well to try and understand and make a link on these particular patients, and to compare them with our cohorts in the UK with completely different diet and lifestyles. And also from the perspective of the Politécnico, we have great support from Dr. Reyes, Dr. Ana Lilia Coria, and Dr. Norberto Dominguez, and also from Cicata, Dr. Paul Mondragon, and from Escuela de Medicina, Dr. Ivonne Olivares, and Dr. Ruben Garcia. And our team actually, I'm not sure, I don't know if Darinka Duran is here, yeah, she's right in the back. So she's actually IPN graduate, and IPN is her alma mater, and she's gonna join us as a postdoc who's gonna learn all the techniques that you're gonna see here. So these are multi-omics approaches that we have established in Oxford. And so the goal is to really have a mirror cohort to start addressing these questions about diet and lifestyle, to really provide information in the context of public health as well. And so, for instance, in here we're gonna be able to have whole genome sequencing, so we can quantify the the mutations of these patients. We're gonna have RNA-seq. We have developed a spectral flow cytometry that I'm going to show in the next slide. This one is actually a 70-plex slide, so where we can quantify 70 proteins in this particular slide. And you can actually see already a TLS, so you can see these T cells, the CD4 T cells. You have these B cells in orange, proliferating B cells colored in white, as you can see here. And then all these plasma cells going in pink, which are going to go in here. And these are all the DAPI cells. So, and we are also curtailing it with functional assays to actually determine about candidates. So what are these T cells and what are these B cells responding, to be able to inform our research teams who are doing cancer vaccines in Oxford Cancer and hopefully also in Mexico. And also in terms of spectral flow, so we have collaborated with industry partners to expand our panels in a 4-laser instrument. So this is actually me in the instrument in the Nuffield Department of Medicine. And we started by just doing a 40-color panel because of the motivation that we didn't have enough cells to actually to work with. We only had 5 million cells from a vial, and then so we thought, okay, we pull everything together. And then we were very satisfied with the performance, and we didn't stop there. So we actually developed 3 more panels that look at T cells, B cells, NK cells, stromal cells. We also curtailed with a functional panel so we can have information in the granular level. Then we put all that information by developing computational pipelines and adaptive pipelines to really mine the data from the bottom up to feed into our linear regression models to actually start asking about predictions of immune landscapes that could that we, in the same way as how we did in HIV, to iterate in terms of therapeutic strategies. And with that, I just would like to thank everyone in the consortium as well, so everybody who has been really supportive from the Mexican side, and we're very excited to actually to start the work, and to you for your attention. Thank you so much.
Gracias. Ok, tenemos 3 preguntas, por favor. Las pueden hacer en español también.
¿Qué tal? Buenas tardes. Doctora, gracias por la presentación. Me había espantado que la tendría que hacer en inglés, pero... es asombroso ver cómo las estructuras linfoides terciarias han ido cobrando relevancia, ¿no? Nada más la veíamos en clase como una estructura que aparece y desaparece y nada más. Una pregunta sería si estas estructuras solo se ven en condiciones crónicas o también lo podemos ver en condiciones agudas. Me imagino en el sitio de aplicación vacunal, si ahí podría estar generando una estructura linfoide terciaria.
Eso es una buena pregunta. Generalmente en la cuestión de vacunación siempre siempre nos vamos contra los secundarios o vamos contra los ganglios. Pero sí, ahora que estamos viendo, por ejemplo, en la cuestión de terapias en donde hay inmunización local o oral en ese sentido, sería muy interesante empezar a buscar ese tipo de estructuras en modelos animales, empezando por ahí. El problema de los modelos animales es que estas estructuras generalmente no se ven. Entonces, es posiblemente una de las razones por las cuales estamos limitados de ver esas estructuras, porque en humanos generalmente tenemos ahora, bueno, sangre y la biopsia líquida que nos da información del ganglio. Pero no, si tuviera que tener una hipótesis, probablemente sí hay una función local que hace sinergia con la respuesta secundaria en ese sentido.
Gracias, doctora. Y ya que logren caracterizar los antígenos que se estén presentando en estas estructuras y se logre un diseño vacunal, ¿cómo podríamos evitar el síndrome de lisis tumoral que se podría presentar?
En cuestiones de off-target effects o en cuestiones de toxicidad.
Off-target effects, veo que son tumores sólidos los que se están presentando. Y, por ejemplo, un efecto adverso de las CAR T cells, se ha visto el tumor lysis syndrome.
Sí.
Entonces, no encuentro todavía cómo diseñar algo para evitar esa gran destrucción tumoral.
Nuestro approach es buscar un neoantígeno que sea específico para el tumor, que no esté expresado en nuestro tejido adyacente. Es complejo. Y sobre todo desde la perspectiva de la misma manera que en VIH, estas respuestas they're shared between self and non-self. Van a ser, las respuestas B van a ser muy, muy pequeñas, los precursores van a ser pequeños, entonces o vamos a tener que tener un monoclonal que sea infundido en los pacientes, o en verdad ser muy estratégicos en los adyuvantes que utilicemos para we engage that B-cell precursor that goes all the way through. Pero sí, es la avenida de los siguientes 10, 20 años de buscar cuestiones únicas.
Muchas gracias, doctora.
Sí. Tenemos una... on the back.
Gracias, felicidades por excelente presentación.
Gracias.
En una de sus diapositivas vi diferentes cánceres, pero yo estoy interesado en el cáncer de próstata y no lo vi.
Cáncer de próstata hemos tratado y es caracterizado como immune desertic. No vemos muchas células inmunes, pero sí vemos células B. Eso es algo que estamos haciendo en Combat Cancer y es interesante porque pues no esperábamos mucho. Pero es bien difícil. Lo que vamos a hacer es montarlo en el 5000K, en el Cinium, porque en cuestiones de autofluorescencia, técnicamente es difícil correrlo en citómetro de flujo para estar seguros de que esas respuestas están presentes, pero hay potencial.
Ahora, también me llamó, yo estoy haciendo inteligencia artificial y usted mencionó que entrenaron un machine learning. ¿Con esos datos ven todos los cánceres?
El machine learning, de hecho, lo aplicamos a nuestro estudio de VIH, porque we were powered, teníamos los features suficientes.
Pero específicamente VIH.
De VIH. Pero el goal es cuando tengamos suficientes datos de estas, sobre todo de las estructuras en donde uno puede empezar a buscar patterns. Entonces, colaboramos con físicos y matemáticos que estamos buscando patrones que estén asociados con funcionalidad, que podamos empezar a traquear más sistemáticamente en los pacientes, como lo hacemos con H&E, pero más sofisticado en el futuro usando estas herramientas. Definitivamente tenemos que usar esto.
Sí, exactamente, es lo que me interesa a mí. Y sobre esto, precisamente le iba a preguntar sobre la base de datos, porque yo estoy interesado y ando buscando datos de calidad y por ahí al final usted mencionó que es algún estudio lo hicieron conjuntamente con México.
Sí.
Esos datos que tomaron, ¿tienen esos datos? ¿Los pueden compartir?
Sí, la idea, y es uno de nuestros goles de la iniciativa, es generar un Data Commons que sea accesible para investigadores, tanto mexicanos como en UK, para generar hipótesis y que haya una generación de conocimiento que dé a más proyectos. Entonces sí, es definitivamente parte de nuestro core en ese sentido.
¿Y con quién, para tener acceso, qué se necesita?
Platicar conmigo. Y con el consorcio también, pero sí, es algo que estamos trabajando en el futuro, sí, definitivamente.
Muchas gracias por su conferencia. ¿Cuál es el papel de las cancer stem cells en el ambiente, el microambiente? Porque sabemos que definitivamente, aunque hubiera una respuesta inmunitaria, sabemos que finalmente cada tumor es prácticamente como el genoma humano. Aunque se llame cáncer de mama triple negativo, cada persona que tiene ese tipo de cáncer va a ser muy diferente y precisamente por el genoma humano.
Sí.
Por ejemplo, la liberación de interleucina 8, de la 1, de la que sea, tiene que ver mucho con nuestro genoma humano. Entonces, cada tumor es prácticamente como una persona diferente, entonces va a comportar muy diferente. Pero mi primera pregunta sería, ¿cuál es el papel de las cancer stem cells en el microambiente del tumor y si ayuda o al contrario, perjudica?
Sí, es una buena. En cuestiones de asociaciones de las estructuras terciarias que vemos en los tumores, no lo hemos estudiado a detalle. Pero sí, por ejemplo, con el Cinium es algo que que tenemos. Sí tenemos marcadores de cancer stem cells en nuestros paneles gigantescos de spectral flow y lo que estamos esperando es tener números suficientes para empezar a hacer correlaciones de números de frecuencias de cancer stem cells con nuestros immune cells. A la segunda pregunta en cuestiones de que cada paciente es diferente, definitivamente, en mi opinión, el campo está direccionado a tener que usar biopsias, hacer whole genome sequencing y bueno, generar neoantígenos y vacunas que sean curtailed. Claro. Para cada paciente. Desde la perspectiva económica, por eso fuimos muy exitosos con mRNA vaccines. Son baratas, son rápidas de generar. Uno puede iterar rápidamente también. Entonces, eso es una inversión que Oxford Cancer está haciendo también para poder tener ese acceso a personalizar. Pero tenemos el goal también es buscar, aun cuando somos diferentes, tener un sobrelapado de neoantígenos que podamos usar como pool para las vacunas de la misma manera que usamos como VIH.
Sí, y en los tumores es muy complicado obtener una respuesta inmunitaria precisamente porque también los tumores esconden el MHC. Y sabemos que el MHC es principalmente la molécula que va a presentar a los linfocitos T citotóxicos, que son los relevantes, como usted nos hizo favor de mencionar. Entonces, hay muchísimos factores que influyen precisamente para que, aunque nosotros podamos tener una respuesta inmunitaria contra el tumor que sea, digo, hay muchísimos, hay más de 200, cada individuo va a responder de forma diferente. Por decir algo, ¿cuántos neutrófilos hay en ese tumor? Por ejemplo, un trabajo de una de mis alumnas que presentó, se ve cómo influye y cada individuo va a tener diferente número de neutrófilos y va a ser negativo para ese tumor.
Sí.
Y fíjense, bueno, lo comento aquí por toda la gente que estamos aquí, que somos mexicanos y que ustedes van a estudiar, tenemos problemas muy fuertes de lo que comemos, de cómo dormimos. La gente que no duerme, sus NKs van a disminuir tremendamente.
Sí, sí.
Si comen pura, tontería, cosas que no nos alimentan, pues tampoco va a ayudar a nuestro sistema inmunitario si no hacemos ejercicio, etcétera. Entonces, todo eso influye, porque ese es el epigenoma. Eso va a influir en el epigenéticamente, así como nuestros genes.
Sí, no,
entonces, es muy complejo el tumor.
Sí.
Pero bueno, le agradezco mucho por su conferencia.
Sí, no,
gracias.
¿Podemos tomar otra pregunta? Sí.
Sí, muchas gracias por la presentación y por... yo quería preguntar, ¿cómo influye? Bueno, primero, si se podría considerar como un tejido linfoide terciario la inflamación crónica por una mala alimentación, por una alteración en el microbioma, que por ejemplo la carne roja secreta o induce una inflamación crónica local. Y si esto, bueno, siempre me preguntan cómo esto le hace o cómo esto aumenta el riesgo, por ejemplo, de cáncer de colorectal o como enfermedades inflamatorias intestinales como CUSI o Crohn aumentan también el riesgo de cáncer, cuando ahí sí habría como un claro, muy probable tejido linfoide terciario, debido a que es una enfermedad inflamatoria crónica. Entonces, digo...
No, hay twofold question, ¿no? La primera pregunta definitivamente es algo que también estamos trabajando, de la progresión de IBS, IBD, Crohn's a colorectal cancer, sobre todo con estas metodologías de spatial transcriptomics. Podemos aprender de esos paths, porque como dice, como dicen, es muy heterogéneo y podemos utilizar eso como información en general. La respuesta en cuestiones de los TLS, en cuestiones de crónicas, ahí es donde nuestro approach depende mucho de la funcionalidad. Ahorita lo estamos caracterizando, pero hay TLS que pueden ser no funcionales, que pueden ser pro-cancer. B-reg cells, T-reg cells, infiltration, no B cells. It goes on the wrong direction. Autoanticuerpos. Y el truco es, la metodología es encontrar los TLS funcionales que nos den la información para hacer las terapias. Y ahí es donde se va a poner interesante de los candidatos que vamos a sacar en ese sentido.
Porque ahí también se me ocurre, ciertos fármacos aumentan el riesgo de colitis microscópica, como los bloqueadores de la bomba de protones o los AINES, o incluso los inhibidores de la recaptura de serotonina, que aumentan la inflamación. Entonces, digo, con este incremento tan abrupto que tenemos de la incidencia de cáncer, no sé si tenga que ver con estos procesos.
Sí. Yet to be seen. Pero sí, completamente inflamación, cómo va a cambiar. Lo que quisiera decir, si tengo tiempo, por ejemplo, es buscar también en cuestiones de calidad y no de cantidad de respuestas. Y en modelos animales ya se ha demostrado, son un par de modelos animales donde dicen que, por ejemplo, las células T tienen respuesta contra cierta microbiota que es beneficiosa para una respuesta antitumoral. Entonces, ahí es, you fish the good thing and then you go back to therapeutics. Yeah. Exacto. Sí, esas son todas las preguntas que queremos hacer con el Hospital General y el Politécnico. Bueno, muchas gracias.
Agradecemos la valiosa participación de la doctora Pedrosa Pacheco y a continuación el doctor Francisco Gutiérrez Galicia, director de posgrado del Instituto Politécnico Nacional, le hará una entrega de reconocimiento de participación en el Congreso Interpolitécnico de Investigación para alumnos de posgrado. Le damos un fuerte aplauso, por favor. Continuamos con la ceremonia de clausura, no se vayan. En un momentito, en unos 5 minutitos. Muchas gracias.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para estudiantes de posgrado. Un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para estudiantes de posgrado. Un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para estudiantes de posgrado.
Tomen asiento, por favor. Muy buenas tardes, investigadores, docentes, alumnos de posgrado, comunidad politécnica. El Instituto Politécnico Nacional les da la más cordial bienvenida a la ceremonia de clausura del Congreso Interpolitécnico de Investigación para alumnos de posgrado. Es un gusto contar con la presencia de nuestras autoridades del Instituto Politécnico Nacional. Saludamos con mucho agrado a la doctora Ana Lilia Coria Páez, secretaria de Investigación y Posgrado. Al doctor Francisco Gutiérrez Galicia, director de posgrado. Al doctor Norberto Domínguez Ramírez, director de investigación. A la doctora Aide Minerva Torres Huerta, coordinadora Comité Científico Área 4. Al doctor Humberto Ríos Bolívar, coordinador Comité Científico Área 3. Al doctor Luis Gerardo Cepeda Vallejo, coordinador Comité Científico Área 2. Y al doctor Jorge Roberto Vargas García, coordinador Comité Científico Área 1. Doctor Francisco Gutiérrez Galicia, director de posgrado del Instituto Politécnico Nacional, sea tan amable en dirigirnos su mensaje.
Bueno, muchas gracias a todos, muchas gracias por estar aquí en viernes, se los agradecemos y también les agradecemos todo el trabajo que han hecho esta semana. Estimados colegas, profesores, investigadores, estudiantes y participantes, es un honor dirigirme a ustedes en este momento para cerrar formalmente el Congreso Interpolitécnico de Investigación para alumnos de posgrado. Durante estos días hemos sido testigos de un evento que ha reunido a más de 2,000 personas entre presencial y virtual, de las distintas unidades académicas y centros de investigación, no solo del Instituto Politécnico Nacional, sino también de instituciones hermanas a nivel nacional e internacional. La importancia de eventos como el de hoy, como el que hoy cerramos, radica en la oportunidad que nos brinda para compartir conocimientos, experiencias y avances en el ámbito de la investigación científica y tecnológica, que se consolida en el país, y este congreso ha sido una plataforma para que nuestros estudiantes de posgrado presenten el futuro de sus trabajos y dedicación a través de presentaciones orales, exhibiciones de pósters y una dinámica de pasarela de prototipos que nos muestran soluciones innovadoras que están desarrollando para los retos que enfrenta nuestra sociedad. Todo ello fomenta el diálogo entre estudiantes, profesores, que generan y establecen relaciones valiosas para futuras colaboraciones y oportunidades académicas y laborales. En el IPN creemos firmemente que la educación superior y la investigación son los motores para un México más próspero, justo, sostenible, y eventos como este refuerzan este compromiso. Quiero agradecer profundamente a todos los participantes, ponentes y organizadores por hacer posible el éxito de este evento. Gracias a su dedicación, este congreso no solo ha sido un espacio de aprendizaje y crecimiento, sino también una plataforma para inspirar a las próximas generaciones de investigadores. Finalmente, los invito a seguir trabajando con la misma pasión y entusiasmo en sus proyectos de investigación y a seguir colaborando estrechamente con sus pares nacionales e internacionales. Este congreso ha demostrado que cuando unimos esfuerzos podemos lograr un impacto positivo en el desarrollo científico y tecnológico de nuestro país. Muchas gracias.
A continuación, procederemos a la entrega de los reconocimientos de los pósters y prototipos, por lo cual solicitamos a nuestras autoridades ponerse de pie. Empezamos con los pósters ganadores. De área 1 reciben: tercer lugar, tectónica salina y su impacto en la exploración e incorporación de reservas de hidrocarburos a la economía nacional. Un aplauso. Segundo lugar, Geoquímica Ambiental de la Laguna de Mar Muerto, Oaxaca, Chiapas, análisis de metales pesados y parámetros físico-químicos en muestras de agua. Y el primer lugar, optimización de inductores de línea para un rectificador trifásico activo. Continuamos con el área 2. Reciben el tercer lugar, evaluación in silico de borosaxolidonas de aminoácidos básicos como inhibidores de enzimas bacterianas para modular la proliferación. Continuamos con el segundo lugar, evaluación de la acción de un SIRNA dirigido a GPR45, GPR82, GPR153 y GPR158, que se está evaluando como un posible tratamiento la lesión pulmonar asociada al cigarro electrónico o al vapeo. Y en primer lugar, identificación de variantes patogénicas en genes involucrados en el desarrollo de estructuras ectodérmicas asociados a displasia ectodérmica en pacientes mexicanos. De área 3 reciben el tercer lugar, gestión y movilidad sustentable en el transporte aéreo, estudio de los grupos aeroportuarios en México. En segundo lugar, integración de energías renovables para una ciudad inteligente, sostenible en países desarrollados, caso de Islandia, Noruega y Dinamarca. Y en primer lugar, estrategias de gestión directiva para el impulso de educación financiera en las estudiantes de bachillerato. Y de área 4 reciben el tercer lugar, evaluación de índice de antagonismo y la generación de enzimas ligninolíticas en un cocultivo fúngico. El segundo lugar, caracterización fisioquímica del colágeno obtenido de residuos de pescado. Y en primer lugar, diseño, síntesis y evaluación del efecto antialodínico de un antagonista de los receptores sigma 1. A todos los que acaban de recibir les brindamos un muy fuerte aplauso. Continuamos con la entrega de reconocimientos a prototipos. Reciben por el nombre del proyecto el sistema portátil e inalámbrico de electromiografía superficial basado en el Internet de las Cosas. Continuamos con el desarrollo de un prototipo de exoesqueleto robótico para rehabilitación de miembro superior. También recibe el ensamble membrana electrodo libre de catalizadores a base de metales nobles aplicados a la reacción catódica en el electrolizador PEM. Continuamos con el secador con sistema de visión por computadora. También recibe el extrusor para biopolímeros. Y finalizamos con el medidor de ángulo de contacto. A todos los que acaban de recibir también, un fuerte aplauso. Sean tan amables en ocupar sus lugares, por favor. Solicitamos respetuosamente a la doctora Ana Lilia Coria Páez, secretaria de Investigación y Posgrado, nos brinde su mensaje y lleve a cabo la clausura de este congreso.
Buenas tardes a todos. De verdad quiero agradecerles muchísimo su presencia el día de hoy, sobre todo, como decía el doctor Francisco, en viernes por la tarde. Así que, bueno, nuestro doble agradecimiento. Al concluir este congreso, quiero expresar mi profundo agradecimiento y admiración a cada uno de los estudiantes politécnicos que han compartido su conocimiento, su entusiasmo y su visión en un futuro mejor. En cada proyecto, en cada presentación, hemos visto reflejados los valores de nuestra institución, la innovación, la excelencia y el compromiso con el desarrollo de nuestra sociedad. Hoy más que nunca queda claro que ser politécnico es llevar en el corazón la responsabilidad de transformar y construir. Y particularmente me siento muy emocionada porque me gusta mucho ser profesora, ser mentora de mis estudiantes. Es un gran privilegio y lo agradezco porque toda la comunidad, a mí y a nuestros compañeros, nos concede que podamos hacerlos y acompañarles. Igual que, pues aunque nos ven ya mayores, también nosotros fuimos jovencitos como ustedes y también tuvimos profesores que en su momento nos inspiraron y nos hicieron sentir que queríamos ser como ellos. Y creo que a toda esta comunidad del instituto que va construyendo día a día lo que somos, pues hay que reconocerle y agradecerle. Y ustedes forman parte de estas personas que en un futuro también serán mentores y también inspirarán a alguien. Fíjense que yo quería tener muchos hijos cuando era joven. Decía, quiero tener muchísimos hijos. Y por alguna cuestión de la vida no pude tener muchos hijos, solo tengo una hija. Pero saben que agradezco mucho a la vida la oportunidad porque pues mi hija se ha graduado, ha estudiado, ha recibido reconocimientos. Pero cada que tengo el privilegio de estar en eventos como este, es repetir esa emoción de orgullo, de felicidad, de ver a los estudiantes aprovechar todo esto que el instituto de manera tan generosa nos da y pensar que todos nosotros somos parte de eso. De verdad que estaba yo luego medio molesta con la vida porque decía, pero ¿por qué hay gente que tiene muchos y yo nada más tengo una? Pero fíjense que también aquí es algo importante para su futuro, ¿eh? A veces nosotros le pedimos cosas a la vida y esperamos que así como las pedimos nos lleguen. Pero yo creo que si somos capaces de ver más allá, de ver más lejos, la vida siempre es generosa y nos da lo que queremos si trabajamos. Y a mí la vida, que ha sido bien generosa, me ha dado la oportunidad de acompañar a muchos jóvenes como ustedes, enseñarles, emocionarme de sus logros. Y hoy, especialmente este día, estoy muy orgullosa de los estudiantes de posgrado del Instituto Politécnico Nacional. Sigan poniendo a donde vayan la técnica al servicio de la patria. Muchísimas gracias.
Es así como damos por finalizado este magno evento, no sin antes agradecer el favor de su presencia. Y en ceremonias tan importantes como esta, ya es toda una tradición levantar la voz de júbilo que une a los politécnicos con nuestro famoso Güelum. Nos ponemos de pie, por favor. A la cuenta de 3. 1, 2, 3. A nuestras autoridades los invitamos a trasladarse a la primera fila para la toma grupal de fotografía de cierre. A nuestro auditorio, por favor.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado, un espacio académico y colaborativo para compartir ideas y experiencias de expertos nacionales y extranjeros. Es una excelente oportunidad para impulsar juntos la ciencia y la tecnología de nuestro país.
Bienvenidos a nuestra transmisión. No olvides unirte a la conversación en los comentarios y compartir este contenido. En breve continuamos.
Soy Francisco Gutiérrez, director de posgrado. Sean bienvenidos al Congreso Interpolitécnico de Investigación para Estudiantes de Posgrado.